This presentation is going to be on cancer biology. So we have tumor suppressor genes, which normally slow down cell division and call for apoptosis. When these aren't functioning correctly, there's the ARF gene, which is found in melanoma, the BRCA2 gene, which is found in breast and ovarian cancer, and the SMAD4, which is found in familial polypsis. There are mutations when it comes to signaling, which is a change in a DNA sequence, changing the amino acid sequence. It can be deadly, either being cancer or sickle cell or something along those lines. So the different types include point mutations, which is substitutions, insertions, and deletions. Chromosomal mutation, which is inversion, deletion, duplication, and translocation. And a copy number of variation, which is gene amplification and expanding trinucleotide repeat. So DNA damages can be spontaneous, and there's two major forms: deamination, which is of the adenine, cytosine, and guanine, and depurination, which is the loss of the purine bases resulting from a cleavage of the bond between the purine bases and deoxyribose. Then there's DNA repair, which can include base excision repair and mismatch repair. There are environmental factors that can influence these damages. One is melanoma from UV radiation. Melanoma is a skin cancer that starts in the melanocytes, which are what give the skin its color. The known environmental factor is sun exposure, especially without protective measures. While the less common type, it is most likely to grow and spread faster. It is uncontrolled growth of these pigmented cells and located in the bottom layer of the epidermis. It arises from a mutation in the MAPK signaling pathway. This pathway is associated with the BRAF proto-oncogenes and the NRAS proto-oncogenes and the KIT proto-oncogene receptor tyrosine kinase. In the BRAF mutation, it is a substitution of valine to glutamic acid at codon 600, which leads to the activation of BRAF. The two tumor suppressor genes involved in melanoma are RB and P53. When these have a mutation, there's unregulated cell growth. And then there's also viruses to tumors, including the Epstein-Barr virus and HPV. So cell growth, there are four main phases. It starts at G1 and goes to the S phase, to the G2 phase, to mitosis, which is prophase, metaphase, anaphase, and telophase. And then there's initiation, progression, and target environments that influence cell growth. In tumors and other proliferating or developing cells, the rate of glucose uptake dramatically increases, and lactate is produced, even in the presence of oxygen and fully functioning mitochondria. This is known as the Warburg effect. There is also pro-angiogenic factors, which include the vascular endothelial growth factors, which is the most potent angiogenic factor, and it stimulates extracellular proliferation, chemotaxis, survival, and vessel permeability. It was originally designated as an endothelial cell-specific mitogen. Its role in angiogenesis includes regulating by inducing the expression of VEGF. Basic fibroblast growth factor mediates a large number of homeostatic functions and developmental factors in different tissues. It is highly potent. VEGF and BFGF require each other to work synergistically in angiogenesis. BFGF requires VEGF to function, and it is important in its expression. Angiopoietins/Ti receptors are important in angiogenesis. It acts as vasculature-specific ligand system to control cell survival and vascular maturation. The deletion of the Ang1 produces a phenotype that is similar to the loss of the Ti2, which is important vasculogenesis to remain intact, but angiogenesis was inhibited. PDGF in genetic ablation inhibits recruitment, resulting in leaky, malformed blood vessels. It also gives an increased risk to EC apoptosis. Finally, overexpression promoted tumor vessel stabilization, whereas inhibition reduced recruitment with an increase in EC apoptosis. And for the anti-angiogenic factors, we have thrombospondin, or TSP1. It binds to integrin and cell receptors, growth factors, cytokines, and extracellular proteases. The molecular scaffold that facilitates between proteins regulating cell morphology, signaling, and adhesion, which possibly promotes receptor clustering. The targeted deletion of TSP1 increases tumor angiogenesis and growth. It has the inability to mount a normal angiogenic response in other assays in mutant mice populations. TSP1 is a direct target of the tumor suppressor gene p53. It is also inversely correlated with progression of carcinomas and melanomas in humans. And then there's endostatin and tumstatin, which are proteolytic cleavage fragments derived from collagen molecules. They influence the effect of the EC integrins and cause apoptosis, suppressing various growth in human tumor cells. Cellular senescence is unique in a way that they eventually stop multiplying but won't die off when they should. They instead remain and continue to release chemicals that trigger inflammation. There's also tumor invasion and metastasis when the tumor begins to spread. So biomarkers are a biological molecule found in blood, bodily fluids, or tissues that is a sign of normal or abnormal processes or of a condition or disease. This image shows the lung biomarkers, which was my organ throughout the course. So there's biomarkers of exposure and biomarkers of effect, as well as biomarkers of susceptibility, which all allow for exposure of the different cells to indicate whether or not the immune cells are reacting correctly to the treatments and everything else that goes along with cancer. So there are different types of treatments, including chemotherapy, hormone therapies, targeted cell cancer drugs, immunotherapy drugs, and bisphosphonates. And for resistance, we have 5 different methods. One is tumor growth that can influence the resistance if it's continuously growing past what the treatment is allowing for. Inadequate drug dosage, suboptimal schedule, drug sanctuary sites such as having to cross the blood-brain barrier, and poor diffusion to cancer cells. And here are my resources.